What's up students, welcome to my channel the Bio Lab. In this video we're gonna discuss about the cell cycle and cell division. Okay, so in this video I'm gonna discuss the cell cycle and cell division, but in previous video we have discussed the topic structure of chromosomes, DNA and other proteins. Okay, so the previous chapter and this chapter lies in the same chapter, or the previous topic or the same topic is lies in the same chapter of ICSE Biology class 10. Okay, so let's start. In this previous video we have discussed about the topic structure of chromosomes, DNA and other types of proteins. So we have discussed the topic regarding the structure of chromosomes, discovery and its definition. Okay, chromatin, DNA, its discovery, formation of DNA and schematic diagram of DNA. Okay, and histone proteins, like other type of proteins I have told that histone proteins other than DNA, genes and why we need reproduce new cells. So I have discussed these 6 topics in previous video. So you should have must watch first the previous video, then for better understanding in this video. Okay, so please watch the previous video first. Okay, so now let's start. What is cell division? Okay, now it raised, what is cell division? In order for cell to divide, it must complete several important tasks, right? The cell must grow, synthesis materials like proteins, RNA and etc. and duplicates its DNA before physically splitting into 2 daughter cells. Okay, so in previous video we have discussed why we need reproduce new cells or why we need cell division. We know, right? For repair, for growth, for replacement, for reproduction, etc. Okay, and now it's needed to synthesize materials like proteins, RNA, and it also needed for duplication of its DNA before physically splitting into 2 daughter cells. Okay, cell perform these tasks in an organized, predictable series of events. So this cell performs in an organized, we can predict the series of events which can be called as cell cycle. Now what is the final definition of cell cycle or the definition of cell cycle? So the definition of cell cycle is, the cell cycle is a series of events that takes place in a cell leading to duplication of its DNA and subsequent division of the cell to produce 2 daughter cells. Okay, so it is a series of events that takes place in a cell leading to duplicates its DNA, right? The point of time where the cell leading to its duplication of its DNA, the DNA is the replication of DNA or the duplication of DNA where the DNA is replicated and the subsequent division of cell to produce 2 daughter cells. Okay, now in cell division there are 2 types of cell division. Okay, that is mitosis and meiosis. Okay, so every cell division there is a preliminary round of the first steps or the preparation for cell to divide, and the first step is interphase. Okay, the interphase, the 2 daughter cells produce from a mother cell are relatively small with a full-sized nucleus but relatively little cytoplasm. These cells are said to be in interphase. Okay, 2 daughter cells that is produced from the mother cells are relatively small. We know that, with a full-sized nucleus, but a relatively little cytoplasm. With a full-sized nucleus but relatively cytoplasm. Okay, so in this event or in this phase we can say that The cell is in the interphase. During interphase, they prepare for the next cell division and grow to the same size as their mother cell. So in this phase, they prepare for the next cell division. Okay, right? So interphase itself has 3 phases. Okay. So what is the first phase? The first phase is first growth phase, which can be said as G1, which can be said as G1. Okay, the first growth phase. Second one is synthesis phase. Okay, synthesis phase, which can be determined as S phase. Okay, and the third growth phase is second growth phase. Okay, G2. Okay, it can be determined as G2, second growth phase. Okay, so here is the diagram of the cell cycle. Okay, so this is mitosis or meiosis phase, and basically it is the mitosis phase, but it is G0 I will discuss later. So it is the G1 or the first growth phase. Okay, so it is the G1 first growth phase. Okay, so this is the first growth phase G1, and synthesis phase. Okay, so this is the synthesis phase. Okay, region DNA replication we have talked about later, but this is a synthesis phase. Okay, synthesis phase, and the second growth phase is G2. Okay, second growth phase is G2, and it is mentioned here. Okay, so this is the first growth phase. This is called synthesis phase, and the second growth phase is G2 here. Okay, so next move to the next topic. First growth phase G1. Okay, the RNA and the proteins are synthesized and the volume of cytoplasm increases. In this phase, the RNA and proteins are synthesized and the volume of the cytoplasm increases. The mitochondria in animal cells and chloroplasts in plant cells are divided. The main idea to withdraw from the cell cycle and entering a resting phase. So in Z0 phase, we have talked about Z0 phase is called the Z0 or the resting phase, R, also called the R phase, known as resting phase. Okay, or the start of preparing for the next division by entering the next synthesis phase. Okay, whether after the division of the cell cycle, it will again go to the rest. It will go to the rest phase, or we can say that R phase or Z0 phase, or again it start preparing for the next cell division by entering the next synthesis phase. Okay, right? So now synthesis phase, nothing. More DNA is synthesized, the chromosomes are duplicated. Okay, DNA replication, so chromosomes are duplicated. Simple. In this stage, the synthesis phase takes place. Okay, for cell division. Now the second growth phase G2. This is a shorter phase in which RNA and proteins is necessary for cell division continue to be synthesized, right? Okay, so in shorter phase in DNA, in a phase in which RNA and proteins, other proteins like RNA histone proteins and necessary for cell division to continue to be synthesized, means the other types of proteins like histone proteins and RNA are continued to be synthesized, will be synthesized in this second growth phase. Okay, in this phase for cell division. So now mitosis. Okay, so mitosis is a cell division. Is a type of cell division in which one parent cells divides into two identical daughter cells by same normal chromosome number. Okay, same normal chromosome number is maintained at each cell division. Okay, mitosis is cell division in which one parent cell divides. Only one parent cell is divided into two identical daughter cells. Okay, one parent cell divides into two identical daughter cells, but the same normal chromosome number is maintained at each cell division. Okay, so now it is the parent cell, it divides in this process and it maintains, see, the same number of chromosomes. See, it maintains the same number of chromosomes and the nucleus. Okay, so here is the process. You can see that here is the stages for the mitosis phase. So interphase, the first phase, now then prophase, where in interface the cell grows, performs routine life processes and prepares to divide. As we know that in prophase, chromosomes are visible. See, the chromosomes are visible. Nuclear membrane breaks down, spindle fiber begin to form. I will later discuss later in this video only, but it is a short. Okay. Metaphase, sister chromates line up at the center of the wall. See, it is lined up. Anaphase, sister chromates are pulled apart and move to opposite folds of the cell. It is like the chromosomes are splitting in from the middle, and it moves towards the opposite fold. Okay. And telophase. Telophase, spindle fibers break down, nuclear membrane forms, and chromosomes begin to uncoil and form chromatin. Okay. The cytokinesis, cytoplasm splits into two daughter cells, means the cell division of cytoplasm. Okay. So let's move to the karyokinesis. Okay. So here there is two types of cell division in mitosis. That is one type of karyokinesis and cytokinesis. Okay. Here is cytokinesis, and karyokinesis means it is the division takes place in nucleus. Okay. So remember it, students. Karyokinesis means division in nucleus, and cytokinesis means cell division in cytoplasm of the cell. Okay. So in karyokinesis, this cell division has 4 phases of mitosis. Okay. In karyokinesis, the cell division has 4 phases of mitosis. That is prophase. Okay. So prophase we have discussed. Metaphase. Okay. Anaphase. And guess what? Telophase. Okay. So prophase, metaphase, anaphase, and telophase. These are 4 phases of mitosis in karyokinesis. Okay. So let's discuss the prophase first. Okay. Now the prophase. Chromosomes have become short and thick and clearly visible inside the nucleus. Okay. So you can see that the chromosomes becomes short and thick and clearly visible inside the nucleus. Okay. Now each chromosome has already duplicated to form chromatids. Okay. Each chromosome is already duplicated in the interphase stage to form 2 chromatids, right? Okay. This one chromatid, one sister chromatid, and this is another sister chromatid, right? Okay. So the 2 sister chromatids, okay, it contains the 2. One chromosome contains the 2 sister chromatids, and others are same as, and the 2 sister chromatids remain attached to each other at a small region called centromere. Okay. So in this point we can see that the middle portion of the chromosome, like 2 sister chromatids are attached in the middle of the chromosomes, are called centromere. Okay. And the centrosome split into 2 along with simultaneous duplication of the centrioles contained in it. Okay. The daughter centrioles move apart and occupy opposite poles of the cell. Each centriole is surrounded by a radiating rays termed as aster. Okay. So the centrosome, here the centrosome splits along with the simultaneous duplication of the centrioles contained in it. Okay. The daughter centrioles move apart. So the centrosome duplicated into the 2 centrioles, and the centrioles, daughter centrioles, it is the daughter centrioles and the daughter centrioles, 2 daughter centrioles are moved apart in the opposite poles. It is a moving, like it is a moving and opposite poles, and this structure is actually the spindle fiber we discussed later about in this video, but the daughter centrioles move apart and occupy the opposite pole. Here one daughter centriole will move up and another daughter centriole will move down. Okay. So it forms like an opposite two, like in the earth north pole and south pole. Okay. Now the opposite poles of the cells, and the each centriole is surrounded by the radiating rays, and it's termed as aster. Okay. So now the centrioles, daughter centrioles, when they move apart and occupy the opposite poles of the cell, these centrioles, these daughter centrioles, are now termed as asters. Okay. Now the number of fibers appears between the 2 daughter cells centrioles and called spindle fibers. Okay. Now I have told this structure. Now some number of fibers appear between 2 daughter cells. It will like this structure, and it will move up, and this type of structure in the two daughter centrioles are called spindle fibers. Okay, spindle fibers. Here you can see this, it is a spindle fiber. Okay, now the nuclear membrane and nucleolus disappear. Obviously, for solution, for the division of the splitting of the chromosomes, right, the nuclear membrane and the nucleus should be disappear. Okay, now how can the chromosome can divide, right? Okay, so duplicated chromosomes start moving towards the equator of the cell. Now the duplicated chromosomes start moving towards the equator of the cell. Okay, now towards the equator of the cell we have discussed, and this the centrioles have discussed that the two daughter centrioles are moved to the opposite poles. Okay, and duplicated chromosomes start moving. It means one chromosome to the another pole, and another chromosome and another pole. In means it is form as a metaphase stage. We will discuss in this video next to the, okay, means prophase, metaphase. Okay, so now we are gonna discuss the stage of metaphase. Okay, so now the metaphase, it will line up like this. Okay, this now the spindle fibers, and see, this is the spindle fiber, and the chromosomes are lined up like this, and this lined up a plate form like structure called metaphase plate. Okay, so duplicated chromosome arranged in equatorial plane. Okay, it arranged in a equatorial plane. Okay, called metaphase plate. Okay, each chromosomes get attached to a spindle fibers by its centromere. Okay, so each of the spindle fiber, as you can see, these are the spindle fibers from the two daughter centrioles or the asters. The spindle fibers attached to its centromere of each of the chromosomes. Okay, and the chromosomes lies on the middle or the equator plane called the metaphase plate. Okay, now the anaphase. Now the main part, the actual main part of the cell division mitosis. Okay, the centromere attaching the two chromatid divides, and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. Okay, the centromere attaching the two chromatids divides, and two sister chromatids to chromosomes separate and drawn apart towards the opposite poles. Okay, now the what happened in this phase, that is, as you can see that we have discussed the metaphase, and these chromosomes are lined up in equatorial plane called metaphase plane, and from this it is attached to the centromere. As you can see that every spindle fiber from two daughter centrioles is attached to each of the chromosomes in centromere, and now from centromere two sister chromatids from two sister chromatids are splitting off from the equatorial plane. Okay, so they pulls towards the each of the opposite poles. They pulls, it pulls. Okay, let me show you. And means it pulls opposite poles like this, and this pulls like this. Okay, and it is the middle point, it is the equatorial point. Okay, so it is the equatorial point, and this pulls this centrioles pulls in this direction, and this centriole push this direction, all these asters. Okay, from the equatorial plane, which causes the splitting of sister chromatids. Okay, splitting of chromosomes by sister chromatids. Okay, and the sister chromatids from the chromosome, they move towards each of the opposite poles. Okay, the drawing action. Okay, the drawing action is performed by the contraction of spindle fibers. It acted that this action, the drawing action, is caused or performed by the contraction. They contract like this, and there is this aster also contract like this. By the performed, this action is performed by the contraction of spindle fibers, right? Okay, now the last phase of the mitosis. Okay, and that is telophase, right? Okay, each chromatins or the daughter chromosomes lengthens becomes thinner and turns into a network of chromatin threads. Okay, now the last phase, the cell has successfully divided, means no, sorry, the cell, not the nucleus, is successfully divided, okay, and becomes thinner and turn into a network chromatin threads. Okay, now the daughter chromosomes again the lengthens and becomes thinner. Now, like, it is from the interphase, means like it is completely divided the nucleus, and then it will go as a rest, or it will again go to the first growth phase, and then so on go on, and it will divide more. Okay, and turns into a network of chromatin threads. Okay, now you can see that it is a turns into a chromatin threads inside the picture of this, and it is network chromatin threads which it turn. And the most important thing, in prophase the nuclear membrane and nucleus disappeared, and here after the cell division in mitosis the nuclear membrane and the nucleus reappears in this cell division, or in this picture, as you can see that. Okay, this is a cell nuclear membrane. Okay, so now actually what is karyokinesis and cytokinesis? Okay, nothing. All the nuclear changes that occur during the cell division are collectively termed as karyokinesis. Okay, karyokinesis and cytokinesis means the karyokinesis, we have to learn the first karyokinesis. Nothing so much, the nuclear changes, the nuclear division, the nuclear division of the cell is collectively termed as karyokinesis. Okay, now the karyokinesis is followed by the cytoplasmic division, which is termed as cytokinesis. Nothing. After the division, as you can see that in telophase, after the division of the nucleus in the cell, the cytoplasm also divides. Okay, and this cytoplasmic division in cell, which is followed by the karyokinesis, is called, or is termed as cytokinesis. Okay, so now the significance of mitosis. Okay, why mitosis cell division is required, or why it is needed for our cell division, right? You are curious much to know about it. Okay, so the significance of mitosis are as: first, the growth. Yes, obviously for the growth, and repair of our body, like repair for any injuries, you can repair. Okay, this causes the mitosis. And replacements, like the many million RBC cells are died every second, and new cells are replaced from its dead cells, so it also helps in replacement for new cells. Okay, mitosis is needed. Okay, now the asexual reproduction, like in the amoeba and other type of reproduction, here the mitosis is causes. Okay, now the maintenance same chromosomes number in daughter cells. The most important thing, this is the most important thing. Okay, that is it maintains. Okay, it maintains the same number of chromosome number. Okay, same number of chromosome in daughter cell. Okay, it maintains the same chromosome number in daughter cells. Okay, this is the main and the actual fact of mitosis. Okay, as you can see that in mitosis there is means after the cell division, the in telophase we have seen that in telophase we have seen that the same number of chromosomes are replicated, like this also same number of chromosomes is replicated, and like this also maintenance of same chromosome number in daughter cells. So in both, after the cell division, the same number of chromosomes in daughter cells are maintained. Simple. Okay, Now the meiosis. Okay, meiosis is a type of cell division. It is not that much, but it is slightly complicated than mitosis. Okay, so let's see. Mitosis is a type of cell division that reduces the number of chromosomes to n equals to n plus n. Means the two daughter cells means two sister chromosomes, means it is the 4n means the total the total number of chromosomes, but here you can see in the parent cells by half and produces 4 gamete cells. In mitosis we have discussed that in mitosis the same number of chromosomes in daughter cells are maintained, but here in meiosis cell division, the chromosome number from the parent cells are halved in the daughter cells to produce 4 gamete cells. Okay, this type of cell division can occur only germinal cells. Germinal cells means sex cells, like what? Sperm and egg cells, right? Okay, and better to remind you that in mitosis means the mitosis causes all the somatic cells except sperm and egg cells, and meiosis causes only the germinal cells of the sex cells like sperms and egg cells. Okay, so I have not included the division of meiosis because the division of meiosis is not given as per the latest ICSE syllabus, so I am not mentioning it in this video. Okay, later on the higher classes you can learn. Okay, so now the significance of meiosis. The chromosome number is halved. First n plus n equals to 2n, and it will be 2n plus 2n equals to 4n, means it is total number of chromosomes, but it is halved. 2n means the total number of chromosomes is halved into two different number of chromosomes contained in each of the daughter cells. Okay, the next is the maternal and the parental chromosomes get mixed up during the first reduction as the separate from the homologous pair. Okay, the maternal and the parental chromosome get mixed up during the first reduction as a separate from the homologous pair. Okay, the maternal and the parental chromosomes, are get mixed up. Okay, get mixed up as a separate from the homologous pairs. Okay, the chromatin material very often gets exchanged between the two members of homologous pair, which can be termed as crossing over. Okay, now the chromatin materials in the cells which very often gets exchanged means the maternal and the parental, from the mother and the father, the chromosomes which are gets bit exchanged, that their gene and the DNA gets the material, the genetical material gets exchanged between the two members of homologous pair, which causes the actual the mixing up of the two different parents, mixing up of two genetical materials of two different parents. Okay, in homologous pair, which can be termed as crossing over. So let's see what is crossing over. So crossing over is the exchange of genetic materials. So it is the exchange of genetic materials between the two members of pairs of homologous chromosomes. Okay, so it is the exchange of genetic materials between the two members of a pair of homologous chromosomes. So this is like we can see that we can say that this is let's see, this is maternal, okay, or this is mother cell, or this is father chromosome. Okay, this is mother chromosome and this is father chromosome. Now in this case, the two homologous chromosomes of maternal and parental, or mother or father. Okay, now when when the meiosis cell division, the chromosome inside the nucleus are crossed over. Okay, are crossed over, and recombinant chromatids are formed. Means non-recombinant chromatids are written here. Okay, not a problem. And here after the exchange of the genetic material, they get different types of, means the maternal chromosome with the maternal material is shared with the father material, or the parental material, or the parental material is shared with the maternal material, means maternal chromosomes. Okay, so they get exchanged. Okay, so the genetic materials of them are exchanged, and this is the homologous chromosomes, as you can see. So this is only the crossing over, nothing so much. Okay, so now thanks for watching. Please like, share and subscribe the video. If you like this video, please subscribe and share my channel and keep me supporting. Love you students. And in next topic, this is means in this video we have completed the chapter Structure of Chromosomes, Cell Cycle and Cell Division. In previous video I have discussed the structure of chromosomes, but in this video we have discussed the cell cycle and cell division. So in total we have discussed the whole chapter 2 of ICSE Biology last time. Okay, so bye for now. And most important thing that in next video we start our new chapter, that is fundamental of genetics, heredity. Okay, so please subscribe my channel, like, share to your ICSE friends and CBSE and other boards also, for this is very informative, as you know. So subscribe and stay tuned, and please turn on the bell icon so you will get the video as soon as possible I uploaded it, latest video. Okay, so thanks for watching, keep supporting me, love you all students.